Hello, everyone, and welcome to Nature and Science Podcast, where science does care what you believe. I'm your host, Don Murphy. We explore the science behind the latest technological developments, ask the deep questions about existence, and seek verifiable truth, leading to a clearer view of reality. On this episode, we speak with Dr. Jonathan Zehr. He and his colleagues discovered an example of evolution in action. Dr. Zehr will share with us how such a revolutionary discovery was made. Jonathan Zehr is the Distinguished Professor at the University of California, Santa Cruz Institute of Marine Sciences. He and his colleagues have discovered the possibility of a complex cellular organism with a nitrogen-fixing organelle derived from endosymbiosis with the nitrogen-fixing bacteria. This new organelle is called a nitroplast. Studying a marine alga with the cyanobacterial endosymbiont, Dr. Zehr and his colleagues, they use soft X-ray tomography to visualize cell structure and division of the alga, revealing a coordinated cell cycle in which the endosymbiont divides and is split evenly, similar to the situation of plastids and mitochondria in cells. In other words, instead of having a symbiotic relationship, the alga and bacteria integrated to form a newly evolved organism. Dr. Zehr, welcome to the program, and we can't wait to hear more about this revolutionary discovery.
Hi, it's a pleasure to be here.
Well, let's start off with what started you on this journey that you're on in science.
Um, so the journey actually started many years ago, back in the 1990s, when I was an oceanographer and I was studying nitrogen cycling in the oceans. Nitrogen cycling in the oceans is important because it controls the productivity of the microalgae in the oceans, which in turn controls the global greenhouse gas carbon dioxide in the atmosphere. So it's important, and I was studying this process, and I was looking specifically for organisms that could do what is called nitrogen fixation. Nitrogen fixation is the reduction of the gas nitrogen, which is in the atmosphere. It's most of our atmosphere, about 78%, but it's biologically inert to all organisms except for a relatively few number of taxa, but diverse taxa that can take that nitrogen molecule in the gaseous form and convert it into biologically available ammonium. Now I'll stop here and just ask if that is clear enough. But those were the organisms I was looking for in the 1990s. That's the start of the journey.
Right. So, in your youth or in your background, what got you interested in science in the first place? Were you always interested in science, even as a kid?
I was always interested in science as a kid. I didn't know that I would get that involved in science, but I was always interested in science. And in fact, my grade school teacher used to give me extra time to go to the library and look at the science books while everybody else was doing their homework.
Right.
So I've always had this interest or ability in science, I guess, curiosity.
Yeah, I was going to ask about the curiosity aspect because we're always trying to get young people interested in science, and that's certainly one of the reasons I do this podcast, not just for young people, but also it started out trying to promote science literacy amongst seniors. To be honest with you, in the
Yeah.
expat community that I lived in Ecuador, because there were so many people that were still afraid of science, or when they talked about science, it was in the context of conspiracy theories. So I was just wondering, as a youth, there must have been a natural, of course, curiosity or proclivity to ask questions and to want to know why things were the way they were.
And I guess I always liked understanding how things work, be it the car engine
Right.
or how bugs fly around or how they eat or whatever. So I guess I was always curious and always wanted to know, you look at these plants and animals, what do they really look like when you can look at them? So I guess I always was just kind of curious to see what things were like and how they worked.
Right. So let's circle back to what you were saying about your journey in science and looking at nitrogen fixation. You mentioned that it was related also to carbon dioxide in the ocean, which is a greenhouse gas, of course, and important. So, what exactly is the relationship between nitrogen fixation and carbon dioxide? If you could just explain that briefly.
Yeah, I'll try to keep it brief. I mean, nitrogen is a nutrient that is important in all life. It's a major component of amino acids and therefore proteins as well as DNA. So all organisms need nitrogen, be they in the land or the sea. We're most familiar with crops because we, of course, are reliant on them to eat. And crops are relying on nitrogen fixation too. They're just relying mostly on us to use basically to burn fossil fuels to do the reduction for us. But also there are bacteria that people have known about for years, some of them some symbionts that live in the roots of some plants, legumes most famously, that do the same thing. They fix nitrogen from the air and self-fertilize themselves with nitrogen. So it's important in regulating the growth and therefore the photosynthesis of any plant. Um, and in the oceans, the plants are microalgae. They're single cell, mostly single-celled algae, aside from the algae that you slip around on rocks near shore. But in most of the ocean, all of the plants are single-celled algae called phytoplankton. And they need nitrogen, and then they photosynthesize, evolve oxygen, but fix
Mhm.
carbon dioxide into organic matter, which then fish and whales and us all feed on.
Understand. And just so people understand, every time we say nitrogen fixation, we're talking about changing that atmospheric nitrogen to ammonium, which is a way that then biochemical processes within the cell can utilize the nitrogen to make things like proteins and
That's right.
components of DNA and other components in the cell. So
That's right.
that's great. So, but what got you focused on this particular alga that you were looking at, and then the symbiotic relationship, the
Right.
endosymbiotic relationship with this bacterium, this cyanobacteria?
Yeah. Well, first of all, I never knew where this research would lead when I started out. I was
Understand.
looking for nitrogen fixing microorganisms in the oceans. And at that time, molecular biology and genetics was just burgeoning with new methods. And I used a method to go look for the gene that encodes the enzyme nitrogenase that does this nitrogen fixation. It's a very similar enzyme no matter what microorganism you find it in. So I was looking for this gene.
Mhm.
Uh, we thought there was relatively few taxa in the ocean that could fix nitrogen, and I went out looking for this gene to find out if in fact there were actually more, and I did. So I found this gene, and I had no idea that that gene would lead to this story of symbiosis at that time. It was a gene we knew it was from something that looked like, um, well, we didn't know what it looked like, but the gene told us it was something like a cyanobacterium.
Mhm.
And I thought it would just be a typical cyanobacterium at that time. And so the rest of the story is twists and turns where we discovered more and more about this organism without actually being able to see it. We couldn't cultivate it. We couldn't see it under the microscope, but we could tell when it was there by its DNA. And we learned more and more by using a variety of fancy methods in molecular biology and flow cytometry. And as we learned, we found out that there was something very unusual about it. It wasn't just a cyanobacterium like I originally thought. It was a strange cyanobacterium. And it was missing a lot of we eventually got the genome sequence, discovered it was missing a lot of the typical metabolic machinery of cyanobacteria in plants. And then we were really curious what it was.
Mhm.
And so we kept trying to figure out what it was. And there was a woman studying a type of phytoplankton in Japan, not interested in nitrogen fixation. She was just interested in these specific types of algae that form calcareous plates, and they're very pretty. You might show a picture of them when they form the calcareous plates. And about the same year that we discovered that this cyanobacterium was not just a cyanobacterium, but that it was a symbiont with an algae, we realized it was the same algae that she was working on. And she realized that inside her algae was a unique body, which by looking for the gene sequence, we discovered that we were actually studying the same thing, a
So how did that
symbiosis.
exactly come about? How did you um, come to the epiphany that, hey, this is the same thing? She published a paper, you published a paper, and the twain met, and you said, "Aha!"
Yeah, so it was a molecular biology again because we um, had finally been able to do these single cell experiments where we could show that this nitrogenase gene that I'd been chasing for over 10 years was associated with a single type of algae that we could identify by a ribosomal RNA gene sequence. Now, that might sound like a lot of jargon, but all organisms have ribosomal RNA. And if
Right.
you get a sequence of the ribosomal RNA gene, then you can compare it to all others and you can determine what the taxa is. So we determined that this nitrogenase gene of a cyanobacterium was associated with a single-celled algae that had this specific 18S ribosomal RNA gene sequence. And that was the organism she was studying that had that same 18S sequence. So it was unambiguous. Our organism, cyanobacterium, was living with the organism she was studying.
And so then you decided to work together to try to understand something, if something deeper was going on. And what I understand from your paper is that you were trying to understand whether or not this was something really unique in the sense that, hey, maybe this isn't just a symbiotic relationship. This may be
Yeah.
an actual organelle that's producing nitrogen fixation. Is that correct?
That's correct. So we were struggling along without our culture, doing everything we could think of to learn more about an organism we couldn't really see or
Mhm.
culture. And in fact, we even wrote in a paper that we weren't sure whether this thing was living inside the cell or was attached to the cell or it was partially inside. And so we were trying to figure that out. When we started talking to, her name is Kyoko Hagino, and she was studying this one specific life stage of this algae where it was clearly internal. So we began to believe that, in fact, yes, it's internal. And that led to the questions of, well, maybe it's not just an internal symbiont. Maybe it's actually evolving into an organelle. And so we'd been wondering about the possibility. And there's one other example in life of a protist that has taken in a cyanobacteria. This protist is called Pollinella.
Right.
And it's like what then was the third example of primary endosymbiosis of a microbe that then becomes an organelle. And
Right.
so these studies had been done, and we started saying, well, maybe, maybe our thing is like that.
Right. Well, before we go too far, let me make sure our listeners understand what we mean when we say organelle. There are individual little globs of inside of a cell that have functions in and of themselves. The most famous of which, of course, are mitochondria. But there are other organelles. Is that right? Inside cells.
That's right. There are other organelles, most famously the chloroplast, which is how the plants around us photosynthesize. That chloroplast is an organelle. It has a specific function, like the mitochondria has a specific function of respiration. But the chloroplast is just for photosynthesis, and it evolved from a cyanobacteria being engulfed by another single-celled organism, and they then grew together as and evolved into a specific relationship where the cyanobacterium became much more simplified and just became an organelle, a factory for the larger cell.
Understand. And when you say became much more simplified, that is, the cyanobacteria became much more simplified. It lost some of its functions and some of its DNA, which were then taken over by the host cell. Is that
That's
correct?
right. It lost a lot of DNA. The chloroplast now has a very, very tiny genome relative to what a normal cyanobacterium does. And so with those genes lost are many biochemical functions that a cyanobacterium would be able to do, but now the chloroplast is reliant on the cell outside it to do that. And in fact, in that case, many of the genes have been transferred to the nucleus of the eukaryotic cell, and then the genes are used to make proteins, which are then re-imported back into the chloroplast, which is
And you
skipping ahead
mentioned
of what we're talking about.
eukaryotic cells. I want our listeners to understand, and most of them are sophisticated, but they're, basically two types of cells. I mean, there's taxes larger than that, but when we talk about the cells here, we're talking about prokaryotes, which do not have nucleus in their cells, and then we're talking about eukaryotes, which are a little bit more complex. They have a nucleus, and they are, in this case that we're talking about, the alga and the polyana that you talked about, are the host uh, cells, and they're eukaryotes, and they're a little bit more complex.
That's right. The eukaryotes have a complex organization, including all these organelles and differences in their cell walls. And what we call prokaryotes are actually a very diverse group of simple single-celled organisms that do not have a specific nucleus and do not have really any organelles. They have a few little tiny intracellular things, but nothing is organized as a eukaryotic cell. So as eukaryotic plant cells or as eukaryotic animal cells, which we are comprised of.
Right. And so that our listeners can make a connection, particularly with these prokaryotes. I mean, they're in our gut. So, for example, our gut bacteria are prokaryotes. They live there. And so we have a connection with these organisms, and they're extremely important to everyone's life cycle on Earth.
Yes, that's been recognized. The microbes are incredibly diverse, and they're able to do many metabolic things that eukaryotic cells can't do, such as nitrogen fixation, just as one example, but there's
Right.
many other examples. And in the last couple of decades, we've realized that these diverse bacteria are everywhere, including, as you said, in our gut. And they function as what the popular term is a microbiome. And they can have incredible effects on your health, many different aspects of life.
Right. Understand. So, what steps did you begin to take once you made this connection? What steps did you and your team begin to take to determine that an organelle had actually moved, from or that an organelle had actually evolved, and that the cyanobacteria was no longer an individual bacterium, but now had become an
Yeah.
organelle?
But the story changed completely when Kyoko Hagino was able to get a culture, a pure culture that could be grown in the laboratory. Before that, we were trying to pluck individual cells from the ocean or sequence large batches of DNA from the ocean and sift through them to try to look at the genome of this phytoplankton to see if any genes from the cyanobacterium had transferred. We had a lot of trouble doing that, and we never really have been able to show that. But about that time, Kyoko, and she spent 12 years doing this,
I
was
understand.
able to get a pure culture in the laboratory, which I should add anecdotally is in her home uh,
Right.
in Japan, and she was able to get this culture. And we had an opportunity to work together through funding from the Simons Foundation. And strangely, this ha all happened about the same time as COVID. So
Right.
the week that we received the culture, in Santa Cruz, California from Japan, we had about 3 days before lockdown. So we had to keep the thing living on the shelf. My graduate student kept it living in her apartment, and um, we had to pick up 8 months later. But once we had the culture, now that meant, to answer your question, we could look for some of the features of what would distinguish something from an organelle from just a simple endosymbiont.
Right.
And as I said, we were just looking for the gene transfer because we know that happened in chloroplasts and mitochondria.
Right.
But I took on a postdoctoral researcher, Tyler Cole, the first author on the science paper, and he had a lot of experience with physiology of phytoplankton and
Mhm.
genetics and even proteomics. And so his approach was, instead of trying to look for the gene transfer, let's just try to isolate this endosymbiont slash organelle in a pure form and find out what proteins are in it. And by looking
Mhm.
at the protein sequences, you can compare the gene sequences for those proteins to the genome of the cyanobacterium and see whether everything that is found in this body is encoded by the genome in that body. If there
Mhm.
was no organelle development, then what you would expect is that this little body with its, somewhat reduced genome now, makes every protein it needs
Right.
by encoding those genes in its genome. But what Tyler found was unexpectedly, at least, I was interested that this was an organelle, but I wasn't sure that I was ready to believe it. But what he found was a large fraction of the proteins that are inside this body could not be encoded by the genome inside that body. They had to come from outside. And that's exactly what happens in a mitochondria or a chloroplast. And
So, in other
it,
words, the host was producing the proteins that
that's right.
the organelle needed or that this body needed. They were being
That's right.
produced outside and being
That's right.
transferred.
That's right. So they're not necessarily genes that originate from the cyanobacteria, but they're genes that encode proteins that will work in the
Hmm.
cyanobacterium. And so he not only found that, but he found that if he looked at the gene, the transcript sequence, the messenger RNA is made from the genome of the host, the nuclear genome, and then those are translated into proteins, and those proteins are imported into the cell. And so the little detail that's not really insignificant detail, it's an important detail, is that when he looked at the sequences for these proteins that are coming out of the nuclear genome in messenger RNA, they all had a common tag sequence on them,
Hmm.
like an identifier,
Right.
like like a postage stamp.
Right. And that
That
was on the end of the.
that's right. And that also is a characteristic that happens in proteins and in mitochondria and chloroplasts, is that their proteins have a unique identifier that target them to that specific organelle and allow the organelle to take it across its membrane.
So we're
So there are some,
actually, so we're actually watching. I want our listeners to understand we're actually talking about witnessing evolution in action. Is that right?
that's the way I call it, or I call it a snapshot in evolution. And I recently was at the AAAS meeting and was asked that question. And the my answer is that we all of us that have taken a biology class learn about the mitochondria and the chloroplasts and how they have evolved. And we all kind of accept that as a fact, but we have to imagine how that happened.
Mhm.
And this thing, this new nitrogen fixing organelle, is essentially something that's sort of halfway between symbiosis and an organelle. It's a snapshot of evolution in action,
Hmm.
in my opinion.
So, what led you to definitively understand scientifically that this organelle was actually uh, once an independent cyanobacterium.
The gene sequences tell us that. We the first of all, the very first gene that I published back in 1998, if we compared that to all other genes, it was clearly like one group of cyanobacteria, and they
Mm.
were cellular cyanobacteria that could fix nitrogen. So
Uh,
that's why we thought originally it's just a typical nitrogen fixing cyanobacteria. But then ultimately, halfway along the story, we were able to isolate enough DNA purified of this organism that we could get a genome sequence, the entire genome sequence. So we knew every protein that could be encoded by it. And it was a very small genome, but all of the genes had this same relationship. They were all similar to a cyanobacterium of the family of nitrogen fixing cyanobacteria. So it's clearly um, something that has evolved out of this
right,
group of cyanobacteria that can fix nitrogen, unicellular nitrogen fixing cyanobacteria.
right. This just mind-blowingly fascinating because it's just wonderful to be able to see a snapshot of actually of evolution in action. I know, of course, Darwin, described speciation and things that would happen when birds, for example, are separated geographically, but being able to see it on the cellular level. Of course, when Darwin was working, we didn't know about DNA, and we
Yeah,
didn't
yeah,
understand what the mechanism was for speciation or how this happened. And now this
exactly.
this is such a clear example.
Yeah, it is. So it's very these sequences are all similar to a unicellular cyanobacterium that has been studied a lot. There's lots of papers. It's called cyanothecae or cyanothece.
Hmm.
And we know a lot about the physiology of it. And so initially I just thought it was going to be a cyanobacteria like that.
Hmm.
But what it is something that has evolved from that group of cyanobacteria and presumably is continuing to evolve. I mean, we don't really know. It wouldn't have to go any further. This
Hmm.
might be it.
Right,
On the other hand, it may continue to lose genes and get smaller and become maybe
right.
even more efficient as an organelle.
So just to recap, so that our listeners understand just how fascinating this is. I mean, you start out, with an alga that really has a symbiotic relationship with cyanobacteria that is its own um, entity, its own animal, its own thing, and they exist in an endosymbiotic relationship. That bacterium is fixing nitrogen so that the alga can use it and carry on its life's processes. And but what's happened evolutionarily along the way is the that bacterium was absorbed into that alga and actually became something different, became an organelle and became totally integrated with that alga. So now that alga is able to carry on its life processes with this organelle, which is now part of it rather than independent from it.
That's right. Now,
Right.
now we have to talk about it differently. There's one organism.
That's just absolutely fascinating. And so, what do you see as some of the consequences of this discovery and the and understanding of this process? We talked about I think, giving us a deeper understanding of the evolutionary process. But what are some of the other consequences of this discovery, and what conclusions have you drawn as you've done this research?
Well, we're still looking into this. This part is not what you asked about, but we're still looking into the mechanisms that make this organelle work as an organelle. So there's a lot of details, for example, involving vitamin pathways that lock these two things together and make them work and grow and replicate uh, together. So those are probably some of the steps that happened. There probably were certain tipping points where things became dependent on each other in ways that made them force the evolution towards becoming more of an organelle. But having studied nitrogen fixation for a long time, of course, the big question is, um, why hasn't this happened in plants, or can it happen in plants?
Hmm.
And there have been many decades of research and whole centers that have been devoted to trying to work nitrogen fixation into agriculture. And it's been a very difficult problem.
Right.
One, because the genetics are pretty complicated. There's over 20 genes involved in the process of nitrogen fixation and many microbes. And most importantly, this enzyme, this chemical reaction, is extremely sensitive to oxygen, which is all around us.
Mhm.
And worse than that, plants make oxygen.
Right.
So one of the big problems when people thought about plants was, well, you have to control all these genes, and people are making progress in that. But at the end of the day, you have to figure out how to get around the problem that the enzyme itself will be inactivated by oxygen within seconds.
Right. Well, that's it's interesting that you're mentioning this, and I just want to interject that I just interviewed the CEO of a biotechnology company, Pairwise. It's a plant genetics company, and they develop different kinds of plants. They just changed the mustard green into a salad green, and they just developed a cherry without pits and that sort of thing. This is in the Research Triangle here in North Carolina. So, and I know this is something he was thinking about as well as how to do nitrogen fixation. And you're saying one of the obstacles to that, which is, I think, fascinating biochemically, and I know I'm being nerdy about this, is the environment where where oxygen is so prevalent, it makes this very difficult. We ran into the same problem, and I hope you'll forgive the digression, when scientists that I just had on the program recently were trying to develop the polymerase chain reaction into something that was a technology that was usable. It was very heat sensitive. Um,
Mm.
and they had to find a bacterium, which they found in national parks where I worked in Yosemite or in Yellowstone. They
Mhm.
found a heat resistant form of another enzyme that then allowed them to be able to
Right.
use the polymerase chain reaction. So it sounds like you're going to have to be able to do the same thing. How will you find something that can operate within this oxygen rich environment in plants?
Well, so that's you asked me what this has made me think about. That's exactly what this is. This is a single-celled plant that's fixing nitrogen. It's evolving oxygen and fixing nitrogen in the same cell.
Right,
So somehow this organism knows the solution that, that we have been musing about for decades.
right,
Um, and so there are things to be learned from how this organism works, either directly possible engineering or not engineering, but somehow the clues to how to allow plants to fix nitrogen without the oxygen inactivation has
right.
been solved.
And it's been solved, and now you're trying to figure out how it was solved.
Well, I'm not a nitrogenase enzyme chemist, and I'm not a bioengineer, synthetic biologist. So that's somebody else's job. And of course, you know, people have been trying to engineer nitrogen fixing plants that or trying to figure out how to do it for, as I said, for many years. At the end of the day, there will have to be some policy decisions about whether or not something like that can be implemented without some sort of biological or ecological risk.
Right, exactly, because that's always a concern, as we've seen with genetically modified organisms. There can always be unintended consequences.
Yeah, and whether or not there are unintended consequences, there are unintended concerns by by people that there might be unintended consequences.
Yes. Well, I'm glad you said it that way. That's That's exactly right. Oh, John, this has been a fascinating conversation. Any final thoughts on this? And I know you say there's a lot of work to be done to look at the different metabolic pathways and the way these organisms actually do what they do. Anything new and exciting on the horizon for you?
Well, we've just nothing I can really talk about in detail, but we have started to do some structural analysis, some high resolution cryo-EM, and so we may be seeing some things at the subcellular level that might be involved in how these processes work. So that's very exciting. And the other thing I'm excited about is just how excited people are about this because it means other people are going to be working on it, and I'm not getting any younger, so maybe I will see some real progress in this.
Right. And when you say subcellular level, and you're talking about function following structure, or structure following function,
Mm.
um, in that genre. So you're looking now on a subcellular level to look at the structure to see if the structure has something to do with how these things work.
Yeah, so one of the things that we found in the science paper was we had done um, this soft X-ray tomography, and we could see the actual alignment of all the organelles, and they're very specifically located. It's not a bag of parts. They're actually very highly structured, and so that probably has some clues about this oxygen problem, about the energy problem. The diagram that you referred to in the Masana article, that shows at the subcellular level how ATP has to be funneled back and forth between the nitroplast and mitochondria, and the chloroplast proximity will affect the oxygen problem, etc. And we know that this intracellular body, this organelle, has its own membranes and it's encased by a membrane of the cell.
Mhm.
So that's a very complicated region that has to do a lot of things. It has to mediate the exchange of all these proteins that are coming from the host to get inside, but it's also got to mediate oxygen. It has to both provide oxygen so the nitroplast can respire, but it also has to control that it doesn't poison the nitrogenase. So the structure of these membranes um, is really important. So understanding what's happening there may be very key to understanding how the organelle works.
Well, I always used to think about that. I know when I was studying photosynthesis, the thing that fascinated me the most was electron transfer and how did electrons know how to go where they go, and
Yeah.
and that's a deeper question than it seems on the surface of it, and I think that has a lot to do with the structure of the subcellular structure that these electrons can follow a particular
Where where
pathway in the.
proteins are located and etc.
Right.
Yeah.
Absolutely. John, thank you for a fascinating discussion. And I can't tell you how much I appreciate you agreeing to join me on Nature and Science podcast. This has been
Um,
great.
hap- Happy to do it.
What a fascinating discussion that was. Thanks for joining me on this episode of Nature and Science podcast. Please don't forget to hit that like button and leave a comment at natureandsciencepodcast.com. As always, stay safe, share happiness, and remember, never stop questioning. Curiosity has its own reason for existing.